Thank you very much for the nice introduction. First of all, I'd like to say it is my great honor to be selected as 2020 Ilchan Memorial Lecturer. As you may know, I'm a cardiothoracic surgeon, and I wonder what should I talk in front of a basic scientist. Usually my surgical society presentations are on the surgical skills or clinical outcomes in which I don't believe you will be interested. So I decided to talk about how I happened to jump into the basic research, and hopefully you can get some ideas on how to collaborate with the clinicians through today's talk of mine. There's nothing to disclose. Lung cancer is the #1 cause of cancer deaths worldwide as well as in Korea. To successful treatment for the lung cancer, it is necessary to collaborate with various specialties and practice with a multidisciplinary approach. Recently, diverse genomic alterations were discovered in lung cancer, and subsequent success of a target agent as well as immune checkpoint inhibitors led translational research to the hottest topic on which basic scientists are willing to work. There would be many reasons why basic scientists try to collaborate with clinicians. Some may just need a sample. However, after today's talk, I hope I can convince you that you need a real collaboration with the clinicians. The subject of my talk will be how to make a good collaboration. The first thing you have to do is finding an academic clinician. Let me talk of my own history. My research interest started soon after I finished my residency program. During the military service, I worked in Seoul District Hospital covering the brew house. As the molecular biology was rapidly developing in that area, which was during my residency program, we were worried about we may stay behind. So to catch up, we organized a study group in the military and studied molecular biology. My second chance of running molecular biology came up during my fellowship at Mayo's Clinic. At that time, I asked a favor to my consultants and was able to take 3 months of molecular biology courses, basic molecular biology courses actually, where I got knowledge of basic techniques, so including DNA and RNA extraction, southern, northern, western blots, or gene cloning and cell culture, and so on. When I came back to Korea, I set up my small laboratories and bought up deep freezer, PCR machines, and other necessary lab equipment by the donation from my wife. And then I started to collect clinical samples and did some benchworks and start publishing several small results. So basically, I followed what the other clinicians and academic position professors typically do. Additionally, I had a chance to collaborate with the NCI, and actually at the time they focused on the GWAS study on non-smoking female lung cancer patients in Asia, and they need a sample. So they recruit many Asian institutions. So my laboratory also sent some samples to GWAS to NCI, and they published several papers actually, including Nature Genetics and Nature Communication and other prominent journals. But however, I was not so much interested in that collaboration because my role in that research was merely a sample provider. Now, in a day in 2011, I was actually driving, and it was when I received a call from Professor Jung Sun-seo, who asked if I have some lung cancer sample. At that time, he discovered a novel mutation gene from a patient with adenocarcinoma of the lung. Let me briefly present the case. The patient was a 33-year-old male never smoked, previously healthy, and no family history of cancer. But he presented with 2 months of history of anterior chest pain, as well as neurologic symptom in his left hand. You can see on the chest X-ray there is a mass in the right upper lobe, which you can see more clearly on the CT scan here. And unfortunately, he had metastasis in the liver, as well as in the cervical spine, and also right supraclavicular lymph node. This is the histologic finding of his lung cancer, and it was poorly differentiated carcinoma, but the TTF1 was positive, so it was adenocarcinoma. But unfortunately, he has no EGFR mutation and no ARC fusion mutation. So what Professor So did is he did the whole transcriptome sequencing using the patient cancer tissue and detected the fusion of KIF5B and RET. And actually, if you look at this slide, there is increased RNA expression downstream of the exon 12, where the KIF5B fusion took place. So at that time, Professor So perceived a novel driver mutation, fusion mutation in lung cancer. But the thing he needs to is to prove whether it is a real mutation or driver mutation or not. So there were several options. One is the traditional way to conduct a functional study, but which will definitely need more time. And the other way is to show it is a recurrent event in the lung cancer. And I think he chose the second one. The issue was how many samples should we scan to get a same fusion gene, which will probably depend upon a prevalence of a KIF5B RET fusion in adenocarcinoma of the lung. We assume that it is probably 2 to 4% of prevalence among the adenocarcinoma, and I opened the sample database and set up a cohort of around 200. I then selected the samples from which either EGFR or ARC mutations were screened and were negative. With such enrichment process, we set up about 55 patients and we performed the RNA-seq for those samples. And as expected, we found additional 3 cases of KIF5B RET fusion mutation. So we reported this to Genome Research, and interestingly, a month later in Nature Medicine, there were 3 independent papers on the KIF5B RET fusion were published. And so we became the first group who reported the KIF5B RET fusion in lung cancer in the world. Through this collaboration, I learned that I could be a part of translational research playing a significant role in the study. Having said that, I appreciate Professor So because a collaboration with him initiated my research works with their basic scientist. So it was a good example of a collaboration between a man with a plan and a man with the stones. We subsequently analyzed 200 cancer patients with the transcriptome and exome sequencing and identified 8 fusion genes on a tyrosine kinase. And we also identified other novel mutations, including 3 cases of MET skipping, exon 14 skipping. And putting them together, we summarized the mutational profile of 200 lung adenocarcinoma cases in Korean population and published it in Genome Research. Meanwhile, I collaborated with Professor Yeol-Hong Kim and Macrogen and started developing lung cancer panel. At that time, I wondered why Professor Kim chose me to collaborate with, and soon I realized the reason was I had a good sample. So for the successful collaboration, you have to find a clinician with the samples. But one thing you have to remember is that the sample should be in a good quality zone. This is an example I experienced while we are developing a cancer panel. We extracted DNA from 32 FFPE archived in the pathology department and found that the only 2 recent samples were satisfactory for NGS analysis. So on the contrary, when I pulled out fresh frozen samples from our tissue bank, I found that the quality of the sample was outstanding. So using those fresh frozen samples, we were able to set up the pipeline of cancer panel analysis in my hospital. Based on the experience, I collaborated with Professor Jong-il Kim, as well as other lung cancer clinicians in my hospital, including medical oncology, pathology, and bioinformatics, and developed the lung cancer panel. We developed the lung cancer panel and as well as the pan-cancer panel using the fresh frozen tissues of the SNUH and published it. And also, the panel were named as the first cancer panel in Seoul National University Hospital, and they are currently in a clinical service. This is an ongoing clinical trial driven by medical oncology colleagues in my university hospital, where they used the first lung cancer panel to search driver mutations that guides choosing appropriate target agent for the patient. The second requirement of sample is number of the samples. Needless to say, it is desirable if the number of available samples is a lot. I have been collecting samples since 2000, and by the early this year, we have more than 1,200 cases of lung cancer with our cancer, normal tissue, and plasma, and PBMCs are all available. But it is oftentimes necessary to select samples with a specific phenotype or genotype to conduct and design genomic researches. So in our sample database, we integrated the genome data as well as clinical phenotype together. Let me show you the beauty of such integrated database. It was 3 years after we published the KIF5 rat fusion when Dr. Young-Suk Ju visited my office offering to conduct a more in-depth analysis of fusion genes. His idea was that the fusion gene generation mechanism seems to be not a simple mechanism, fusion mechanism, but a more complex rearrangement process. It was necessary to analyze whole genome sequencing to clarify it. However, most researchers at the time failed to study this issue as there were not enough fusion mutation gene samples. However, fortunately, at the time, I was running a first lung cancer panel, so that I had a handful of fusion samples. Based on the cancer panel result, we constructed 350 cases, among which we found 51 were fusion gene mutations present. By analyzing whole genome sequencing of them, we discovered that the majority of fusion oncogenes were generated by complex rearrangement mechanism rather than simple rearrangement, as you can see in this pie chart. These are the examples of complex rearrangements that we found during the study, which varied from chromoplexy, as you can see here, or either classical or balanced chromatopsis. Also, using a sample with the copy number gains, we estimate the timing of the fusion oncogene generation. We found that the timing of the acquisition of fusion genes were much earlier than that of other oncogene mutations. For example, for this patient LUFF76, the acquisition of the fusion gene took place at the age of only 1. We published this data on Cell last year, and I can say that the work resulted from a successful collaboration between clinicians and basic scientists. The fourth requirement of the sample is the informed consent. As you may know, regulations are getting stringent in human tissue usage, and I have been establishing IRB-approved tissue bank with the informed consent since 2000 already. So it is very important to make sure that all the samples are IRB approved and consents are available. Needless to say, experiments are getting more and more expensive, and you may need money. So if you can find a clinician with a sufficient research fund, that would be super. Probably the most critical thing in communicating or collaborating with your collaborators are the communication or discussion. And I recommend you to discuss with your collaborator as frequently as possible. During the cell paper, we communicate with the email, KakaoTalk, and also the web-based learning, web-based meeting. And this continues the communication, keep ourselves to stay on the same page. Additionally, if your partner or collaborator has many other collaborators, that is the green light. While revising cell paper, for example, we were requested to conduct pathologic validation by doing a bunch of immunohistochemistry. However, finding old paraffin block seems to be practically impossible. Luckily, I had already set TMA, which I constructed to conduct another research with our pathology collaborators. And I found that most of the fusion samples were overlapping with that TMA. So we invited Professor Yoongyung Chun from our pathology department, and she quickly performed additional experiment using that TMA. I often get a question about why I do research. There are various reasons, but I found myself enjoying it when I work with my collaborators. I remember the moment when we applied on the cover page of the issue of the Cell. Actually, at the time, we were running out of time, but everybody was spending their time choosing a good picture to use. So, and this is the Professor Chu's art piece, in which he spent a significant amount of time while the deadline of revision was approaching. He found the image in the Internet gallery located in the LA. And actually, I bought that picture, the picture, and actually it is hang in my office now. So although it's not chosen as a cover page, it was one of the most entertaining moments to share with my collaborators. We further expanded the collaborative researches. With Professor Seo, we analyzed the whole transcriptome sequencing of lung squamous cell carcinoma as well as adenocarcinoma, and discovered that we could define immune microenvironment signature with the RNA-seq clustering. This is the collaboration with Baylor, where my PhD student is working, and using a similar concept, we proved such immune microenvironment might be useful to predict immune checkpoint inhibitor responsiveness. Professor Na in my department and Professor Choi in nuclear medicine department in Seoul National University Hospital pulled out all the lung adenocarcinoma RNA-seq data and analyzed them. And they found that the differential expression of glucose transporters between cancer cells and immune cells and their spatial distribution can cause reciprocal glucose metabolic competition, and this can be used to predict immune checkpoint inhibitor responsiveness. We published it on the Theragnostics. Using the same RNA sequence data, we conducted a more clinically applicable study by incorporating PET-CT and deep learning algorithm, which may be useful to predict immune checkpoint inhibitor again. And this paper was also published in Theragnostics in this year. This is unpublished data, which is under final step of review in cell stem cells. And my laboratory and KAIST team have been collaborating to set up a lung cancer organoid. Meanwhile, the COVID-19 pandemic took place. Although lung cancer organoid is still not fully established in our laboratory, we have been successfully set up the normal human lung organoids. Collaborating with several other research teams, and we established human alveolar type 2 cell organoid infection model for COVID-19 virus. As you can see on the slide, we took an electron microscope photograph, and you can see there on the C, there is particles of viral particles very well. Also, we performed the RNA-seq for those organoids, and we were able to identify that viral expression reached the plateau within one post-infection, while the type 1 interferon signatures were increasing starting from the first day and reached the plateau at the third day post-infection. So there are many interesting findings on this paper, and which you will probably enjoy in near future once the paper gets published. Please stay tuned. Anyway, it was another good collaboration to set up an infection model system using a human organoid, which can be used in the future research against the COVID-19. So I can summarize that to make a good collaboration with the clinicians, you will need to be a person with a good idea. And you have to find a person with the samples, with a good number, good quality, with a genotype, and informed consent to be ready. And please find the person with the resources, and find a person you can communicate with very well. And you have to find a person who are prepared to work with and enjoy with you. And you should be skillful in communication. Above all, I strongly believe that multidisciplinary collaboration is critical in the current era of translational research. These are my current collaborators, including Professor Zhu and his colleagues in KAIST, and Dr. Lee in Harvard, who wrote most of the cell paper with Dr. Park in KAIST, and Professor Jong-il Kim in GMI, and who have been very helpful to conduct the research, especially the cancer panel researches, and all the members of my department and laboratory. And the most importantly, special thanks to Professor So, who initiated me to collaborate research and taught me how to collaborate. Again, it is my great privilege to be selected as a Ilchon Memorial Lecturer, and it was an honor to speak to basic scientists. Please stay safe and healthy so that we can collaborate in the future with you. Thank you very much.